Altered alveolar macrophages function, the role of smoking. Alveolar macrophages, AMs, are immune cells originating from the bone marrow, initially as peripheral blood monocytes. They migrate to the lungs and transform into AMs under the influence of interferon gamma and lipopolysaccharide, LPS. AMs play a crucial part in lung immunity and inflammation. Complement receptors play a vital role in their defense mechanisms, involving the production of chemokines like interleukin-8, IL-8, monocyte chemoattractant protein 1, MCP1, and growth factors. AMs also regulate adaptive immunity through actions such as producing reactive oxygen and nitrogen species, ROS and NOS. These are the morphology of AM. Smoking typically refers to the act of inhaling and exhaling the smoke produced by burning tobacco. This behavior is most commonly associated with the use of cigarettes, cigars, or pipes.
Going through the prevalence, cigarette smoking among Malaysian teenagers has witnessed a decline. The NHMS survey shows that the rate of current cigarette use among adolescents aged 13 to 17 dropped from 13.8% in 2017 to 6.2% in 2022. By gender, 10.8% of adolescent males currently smoked cigarettes in 2022, compared to 1.7% of adolescent females. In Malaysia, the smoking rate for men is 1 in 55, and for women, it is 1 in 135. Smoking is known to increase the risk of lung cancer. This is largely due to the fact that cigarette smoke contains around 4,000 components, 55 of which are known to be carcinogenic or cancer-causing. Studies have shown suppressed anti-tumor activity of AMs from smokers with lung cancer compared to current non-smokers with the disease. This means that they are less effective at fighting off cancer cells, which can lead to an increased risk of lung cancer. Cytotoxicity levels were reduced in AMs from current smoking patients with lung cancer, compared to non-smokers and ex-smokers. One of the most significant carcinogens found in cigarette smoke is a nicotine derivative known as NNK for short. NNK has been found to downregulate or decrease the production of cytokines by AMs. Cytokines are small proteins that are crucial for cell signaling. In addition to downregulating cytokine production, NNK also inhibits the release of tumor necrosis factor, TNF, and interleukin-12, IL-12, both of which are important for immune response and inflammation. Conversely, NNK increases the release of interleukin-10, IL-10, and prostaglandin D2, PGD2. IL-10 is an anti-inflammatory cytokine, while PGD2 plays a role in sleep regulation and smooth muscle contraction. The combined effects of NNK promote the differentiation of AMs from the M1 to the M2 phenotype. M1 macrophages are associated with inflammation and destroying harmful pathogens, while M2 macrophages are involved in wound healing and tissue repair. However, the M2 phenotype is also known to contribute to the development and spread of cancer. There's a study about the phagocytic capacity and receptor expression in AM by comparing a healthy person and lung cancer patient. In a healthy condition, AMs take up antigens for processing and present them on major histocompatibility complex 1 and 2 molecules. This process typically activates T cells, leading to phagocytosis and the release of pro-inflammatory cytokines. While in the lung cancer patient, it starts when AMs undergo changes involving reduced presentation of MHC class II on the cell surface. As there's reduction of MHC class II, there is a decrease in the expression of other important molecules. Hence, AMs show a decrease in their ability to use antibodies to kill cells. COPD is associated with AM's phagocytic function, investigation of AM phagocytic ability, and recognition molecules using flow cytometry. These molecules are produced by our body as part of the immune system. They are expressed on the surface of cells, including AMs. They play a crucial role in the clearance of apoptotic cells, preventing potential tissue damage. However, in the context of conditions like COPD, their activity may be affected, contributing to the disease pathology. The phagocytic ability of AM was significantly decreased in both COPD groups and in healthy smokers. However, phagocytic capacity was better in subjects with COPD who had ceased smoking, compared with those who were still smoking. When patients with COPD cease smoking, it leads to decreased inflammation and reduced oxidative stress in the lungs. Smoking impacts AM recognition molecules and is associated with cyclic adenosine monophosphate (CAMP) increase. The primary function of CAMP is to serve as a second messenger in intracellular signaling pathways. CAMP transmits the signal to various cellular components, influencing processes such as gene expression, metabolism, and cell function. Smoking affects CAMP levels through various mechanisms. One primary way is through the activation of certain receptors on cell surfaces, particularly those that respond to components found in cigarette smoke. Nicotine, a major component of tobacco smoke, can stimulate these receptors, leading to an increase in CAMP levels. For instance, increased CAMP may contribute to impaired immune function, altered inflammatory response, and changes in cellular processes. A recent study showed that cigarette smoke can induce AM polarization towards M2 phenotype in patients with COPD by deactivating M1. Researchers found that cigarette smoke was able to downregulate M1-related genes and upregulate M2-related genes. IFN signaling refers to the signaling pathway activated by interferons, IFNs. When interferons bind to their respective receptors on target cells, they initiate a signaling cascade known as IFN signaling. Cigarette smoke has been downregulating the IFN signaling. IFN gamma is an important cytokine, which can perform several functions important for the development of activated macrophages during inflammation. Chronic exposure of the alveolar space to cigarette smoke could result in a loss of IFN gamma R alpha chain expression on AMs, followed by a downregulation of IFN gamma signaling. One study has shown that the release of MMP12 and TNF alpha by AMs was altered in mice exposed to cigarette smoke. MMP12 secretion from mouse AMs was increased after cigarette smoke exposure, whereas TNF-alpha secretion showed an initial time-dependent increase and subsequent decrease over 10 days. Angiogenesis is the process of forming new blood vessels from pre-existing ones. In the lungs, angiogenesis plays a crucial role in normal alveolar development, injury, and repair. The initial purposes of angiogenesis is to improve blood flow and stop cancers from growing by choking off the blood supply and starving the tumor. However, exposure to cigarette smoke has been shown to dysregulate many of the functions of the AMs, including angiogenesis. AMs in lung cancer that is due to smoking release growth factors, cytokines, and inflammatory mediators, which regulate angiogenesis. AMs can actually promote the progression of tumors by helping the formation of new blood vessels by process of angiogenesis. ROS, which is reactive oxygen species, and NOS, which is nitric oxide synthase, can be defined as important molecules involved in immune responses and defense mechanisms. Initially, ROS and NOS work in alveolar macrophages to regulate immune responses and combat invading pathogens. However, imbalances in their production caused by smoking can have negative effects on lung health and immune function. The toxic components of cigarette smoke can significantly increase the production of ROS by alveolar macrophages. Excessive production of ROS can result in oxidative stress. ROS, being highly reactive molecules, can damage various cellular components. Over time, the cumulative effects of oxidative stress can contribute to the development of chronic lung conditions such as COPD and lung cancer. In conclusion, smoking has a profound impact on the function and behavior of alveolar macrophages in the lungs. Further research is needed to uncover the intricate details of the relationship between smoking and AMs, so we can pave the way for innovative strategies to combat the detrimental effects of smoking on lung health.